Christy Duncan has been a wonderful partner in our gene therapy world and coming to us from Boston Children's. Thanks so much.
Thank you so much. It's such a pleasure to be here. Sorry. All right. So as we get the slides up, I just want to say it is really an honor to be here and to have been coming for years to see so many familiar faces and great partners that we've worked with over time. I always feel a little funny giving this presentation with just my name up there, since it really has been a community effort from many people, and particularly with Florian, who has been my partner in crime doing this now for the past 6 years, I think. So with that advice and working with my partner, we have changed the objectives today, just thinking about the lunch that is probably getting delivered outside the room as we speak. So originally what we wanted to do is provide an update on cellular therapy overall, so stem cell transplant and gene therapy for children with cerebral ALD. We were going to touch a lot on 2 great studies that have been sponsored by Bluebird Bio, and thank you for help with these slides and the presentation, and focusing on transplant and gene therapy, but we're just going to blow through that first half. If there are questions about stem cell transplant, we've got a table of transplanters over here or others, and I'm really happy to talk about that with you over lunch. We're really just going to focus on the gene therapy side of things. So, but before I do that, I think it's always important, just in different audiences, to talk about why in the world we're talking about a blood therapy or hematopoietic therapy and how that could possibly help a brain disease, because the connection is not natural. And so what we think, and there are proposed mechanisms of effect for cellular therapy, so stem cell transplant using blood stem cells, either gene therapy or stem cell allogeneic transplant, although it's not entirely clear. So this is not an area where the exact mechanism of action is perfectly known. And so the theory is that the cellular therapy mediates correction of the abnormal function of the brain microglia and alters the neuroinflammatory environment. So I think we're going to hear more about the neuroinflammatory environment later on today. So proposed mechanisms, so replacement, which is a debated concept, whether the microglia are actually replaced, or modification of the environment of the host microglial cells by donor-derived hematopoietic myelomonocytic cells. So what does that last part mean? So just for a refresher, or to keep you up to date, so our bone marrow, the hematopoietic stem cells that we're talking about, so sorry to the side of the room, I know you've seen these slides before, many of you. So the hematopoietic stem cells are stem cells that come from the bone marrow. Those cells can then turn into multiple different cells that make your red blood cells, they make your platelets, they make your immune cells. And so what we're doing in this process is taking one of those cells, the circled myelomonocytic cells that come from this baby stem cell, get developed and turn into those other cells, and then pretty amazingly, those cells can migrate into the brain, cross the blood-brain barrier, and then alter the microglia and that environment. So it's really quite a remarkable thing that can happen to help with this disease. And also, we know that inflammation is important with ALD and cerebral ALD, and so anything that alters that inflammatory environment or the immunologic reactions may help with this disease process. So that is sort of the underpinnings of why we think this works. And now, just for a brief moment, I'm going to skip through slides that people have spent a lot of time on. I apologize. And just get to the summary. So this is it's really good data. I promise it will come out. I apologize. So I really just wanted to get this summary. So this is a study, I think, that is ongoing, and it lays the groundwork for why I personally think we need gene therapy in this disease. So what this is a retrospective and a prospective study of the outcomes of allogeneic stem cell transplant. So stem cell transplant where you're getting blood stem cells from another person or cord blood, and how those outcomes fare over time. So what the early data from this study has shown is what we've known, what Florian showed and what Dr. Malik showed us, and I think we may learn more later from Troy and Vinod, others in the room, and that allogeneic stem cell transplant can provide survival and decrease the major functional defects in patients with cerebral ALD if you are treated early in the stage of disease. The neurologic disease progression, measured by your LESS score, which I know is imperfect, and the NFS can stabilize post-transplant. But while allogeneic stem cell transplant works, there are a lot of problems that come along with this. There are practical issues that are not discussed here, but then there are complications. So rates of graft versus host disease. So graft versus host disease is an allogeneic complication. So what this means is that the immune system that you've gotten, these new cells from the donor that are helping you with this disease, they can also attack your body. And so that can cause problems that are minor, like a rash, or they can cause problems that are life-threatening, such as lung disease that requires lung transplantation or even death. So we would like to eliminate that risk of graft versus host disease from the lives of children with cerebral ALD. Other issues are graft rejection. So you go through this process, you get a transplant, and your body doesn't accept those cells, or it later rejects those cells, and that's an issue with stem cell transplant that we need to work on. So in this study, also mortality. So stem cell transplant is an extraordinarily intense process. In the data that you didn't see, or you saw them fast forward, at 1 year, there's treatment-related, not disease-related, but treatment-related mortality of close to 15% in children who undergo allogeneic stem cell transplant. So the summary of that data and that work that is ongoing is that I think there is an unmet need for something different to try to address these issues in boys with cerebral ALD. Which brings us to the Starbeam study. We presented this data last year. There's more data that's come from it, so we'd like to provide an update as to where we are. So gene therapy, for those who aren't familiar, what we're doing in gene therapy and gene addition therapy is inserting genetic material that enables the person to create a functional protein required to carry out essential cellular functions. So in this case, the ALD protein. Gene therapy with auto transplant has been investigated, is being investigated as an alternative to allogeneic stem cell transplant. And one of those goals is to avoid the serious risks that we talked about of stem cell transplant. So the Lenti-D product, which is a product that's being investigated in this trial, it is an investigational, as I said, gene therapy product that has an auto CD34. Those are the stem cells, those baby cells that can turn into everything else. So CD34 enriched population that contains cells transduced with this lentiviral vector to encode for ABCD1 DNA. And that is the product that's being investigated. So in this study that's still ongoing, the primary efficacy outcome is the proportion of patients who are alive and free of major functional disability at 2 years after therapy. For this trial, those major functional disabilities have been defined as loss of communication, cortical blindness, tube feeding, total incontinence, wheelchair dependence, and complete loss of, of voluntary movement. And so while there are other things that are clearly important in this disease, this is what's been defined as the major functional defects in this study. The primary safety outcomes, so the proportion of patients who experience grade 2 or higher graft versus host disease. So graft versus host disease, again, the new cells attack your body and cause all sorts of havoc. It is graded on a 1 to 4 scale, so 2 being patients who have a more significant involvement. Secondary and exploratory outcomes, so changes in the NFS, gadolinium contrast resolution, overall survival, and change in that LESS score over time. Additional key safety parameters that are being investigated are the issues of graft failure. So do we give these cells and they not work, or they don't grow within the patient? Adverse events that come from the study. Detection of the replication competent lentivirus, so the vector that's being used to get that genetic material into the cell, and insertional oncogenesis. So this is a schema, and Florian showed this a little bit before, but I just want to dive into it in a touch more detail. And so in this project, the patients are mobilized or apheresed. So we want to get their own stem cells from their bones into their blood and collect it. To do that, they're given a drug called GCSF, and perhaps in some cases an additional drug to help move those cells from the marrow. Those cells are then collected. The patient is connected to a machine for much of a day, which in some of our young boys can be a little bit of a challenge. Those cells are collected, and those are then sent to a central manufacturing site where the stem cells, so the baby cells that turn into everything else, are selected out. They're treated with the lentiviral vector containing the DNA that we're trying to get into the patient, and then they're stored. They undergo a series of release testing to make sure the cells meet criteria to release to the patient. And at that point, the patient is admitted to the hospital to prepare for this process. The patient does get chemotherapy, and that is something I think that it's not always appreciated with gene therapy. The reason this is done is obviously these children don't have cancer, so why are we giving them chemotherapy? We need to make space in their bodies to accept the cells coming in and give them an advantage. So the child's admitted to the hospital, gets relatively intense chemotherapy for a period of time, and then those stem cells are infused. They are infused like getting a blood transfusion or getting another blood product. So everything surrounding it is important. That process is probably the easiest part of the whole thing, is actually getting the cells into the body. The child then remains in the hospital until they recover. They're released from the hospital and then followed for 2 years on the trial. After 2 years, they're offered participation in a long-term follow-up study for a total of 15 years of follow-up. So right now, I just want to give an update on where we stand as far as the number of patients enrolled and where they are in the trial. Oh, sorry. I guess that means it's lunchtime. And so when we spoke last year, we talked about the enrollment up to that point, and at that point, we were talking about 17 patients. So right now, there have been 33 total patients enrolled on the study. 15 of those patients have gotten to that 2-year endpoint and are being followed in the long-term follow-up program. This slide just shows you where patients are in the process. So almost 94% of patients, as of April, I should say, as of April 2018, where this data, the data cut occurred. So as of April, 94% of patients had had their stem cells collected, 91% of them gone through conditioning, infusion in 88% of the cells, and then 15 of them have completed the 2-year follow-up. There are 4 boys who discontinued, and I want to just briefly mention those patients. So 2 of the patients had enrolled on the study, but during the screening period were determined not to be eligible, so were moved from the study. And 2 of the boys, one of the boys had a rapid progression of his disease. We'll show the data from that child. And the other patient had contrast enhancement after and was removed from the study and underwent an allogeneic stem cell transplant. The boys, the median age was 6 years. At the time. And the follow-up thus far has been 0.4 months to 54 months. And Flora reminded me a couple of weeks ago that in early October was the 5-year anniversary of our first patient getting infused, which is really, I think, quite remarkable and is an exciting milestone for all of us. So how are we doing where the data had matured? So looking at our primary endpoint, so what proportion of the patients are alive and MFD-free at month 24? So this is the data from the boys who reached the 24-month endpoint. Many of the boys in that expansion cohort have not, and so that data is not yet available. So as I mentioned, there are 2 boys who had disease progression. So one child, patient 2018, and you'll see his data quite a bit, had rapid progression after infusion, developed MFDs, and ultimately tragically died of his disease. Patient 2016 had evidence of contrast enhancement that progressed after, decided to remove that patient from the trial. He underwent an allogeneic transplant after that and unfortunately died of complications following that transplant. 15 of the 17 boys who have data available and mature are alive and MFD-free at 24 months. Through 24 months of the data cut point, 54 months of follow-up on this point. And so this is, compared to historical controls of transplant, roughly equivalent to the data that we'd expect to see. As far as the safety outcome, so the number of patients who experienced graft versus host disease, none of the patients experienced graft versus host disease at any point in time. So we look at our following changes in the NFS. The patient who you can see on the top with the rapid escalation, that is patient 2018 who did have rapid change after transplant. The remainder of the boys have continued to have low NFS scores, as you can see following the bottom. 3 of the patients had changes in their NFS from 0 to 1. 2 of those boys had a resolution of that on subsequent evaluation. And 1 patient had an increase from 0 to 2 due to vision impairment and non-febrile seizures. So the less scores over time. And so we can see on the bottom, I know it's difficult to read. This is over time. And we can see, and I'm just going to use my finger over here, I'm sorry. So 24-month time point, you generally see stabilization at the less scores at that point. And this is something we're following closely on this study. And so watching gadolinium enhancement, and I will say in advance, we don't have a full explanation for what I'm about to present. So the rust-colored dots, that indicates a time point where the child on the MRI scan had gadolinium contrast enhancement. As you see, everyone has that rust-colored dot at the beginning that's part of the eligibility criteria for having cerebral ALD in the study. As you see over time, so the markers on the bottom, it's 0, 6 months, 12 months, 18 months, 24 months, and then going out further, that the majority of boys by 6 months had resolution of contrast enhancement. In a subset of the patients enrolled in the study, some contrast enhancement reoccurred at a later time point. In many of those boys, that contrast enhancement has resolved subsequently and is still being followed. What's interesting to note, though, when you look at this, the pictures on the images, that the contrast enhancement that occurred after infusion and after treatment was always less than the contrast enhancement that occurred at the baseline. And so what we see here are just a subset of the patients on the study. They're screening MRI and with the contrast enhancement, and then that at reemergence. So this is something that is being followed and being investigated, and we don't have a satisfactory answer yet. So the safety profile, so this looks at on the left side of the screen, looking at issues related to the gene therapy product. So the drug product, or the vector copy number, is 1.2. The drug product cell dose is listed, and then the time to recovery. So the neutrophil engraftment, this means after you've gotten your chemotherapy, after you've gotten your stem cells, how long does it take for you to start to recover part of your immune system that we measure with this neutrophil number? So the median time was 13 days. If you look below that, you'll see a relatively large range, so 11 to 39 days. That is likely explained by patients very early in the study who did not get G-CSF and medication to help them recover on the trial, and then later time points, later patients did. So I think the median is probably more representative than the range in this case. Platelet engraftment, so the time it takes to recover your platelets, which are the cells you need to clot your blood, occurred at 30.5 days. The adverse events that are recorded, so febrile neutropenia, so having a fever when your blood counts are low, was the most common, followed by stomatitis, which are sores in the mouth, which is also an expected complication of of chemotherapy. Decreased appetite, also expected with chemotherapy, and those being the most common. These side effects, or these adverse events, are all very similar to what you would see in allogeneic stem cell transplant using the same conditioning regimen that the boys received in this study. There were 3 AEs that are considered possibly related to the drug product: 1, a viral bladder infection, 2, elevated heart rates, and 1, an episode of vomiting. So with looking at this data and taking it all in aggregate, we think that the safety profile, particularly based on conditioning and transplant, is very similar to what you would see in boys who had an allogeneic stem cell transplant. So just in summary of the gene therapy data that we have available to date, of the 17 patients who completed 24 months of follow-up, 15 of them, or 88% of them, are free of major functional disabilities. An additional 12 patients have received treatment since that time, and hopefully you'll have us back next year to talk about what's going on with those patients. So we don't have data on the 24 months because they haven't reached that, but to date there's been no evidence of MFDs in those patients, the new addition patients. There's been no graft failure, there's been no graft versus host disease, and no evidence of insertional oncogenesis. So the safety profile is consistent with what we see after transplant. The Lenti-D gene product stabilized neurologic disease progression and may be an alternative to allogeneic stem cell transplant for patients, particularly those with early cerebral disease. And also for patients, and this refers back to the talk you didn't see, who don't have a matched sibling donor. So for patients getting stem cells from another patient, allogeneic transplant, your outcomes are much better if you have a matched sibling donor compared to others. So we think that this, particularly for those boys who don't have a matched sib, this may fulfill an unmet need. Just to follow up, so the study has met its enrollment target, which is fantastic and a huge milestone. Additional follow-up is ongoing, and we're to assess the durability of this response and the long-term safety. So just to close, I talk about Florian and I and our work, but it really takes a global village to make this work. And I really want to thank our partners at Bluebird Bio at the Bluebird table over here who've been really a fantastic team to work with. And we owe so much of the success of this and the future hopeful success for patients to that team. So thank you very much.
Thanks so much, Christy.
I'm hoping we can talk a bit about stem cell science right now. I know a little bit about that. And one of the things I want to clarify with you right now is stem cells is not just one thing. There are many different kinds of stem cells. As I understand it, a certain kind of stem cell transplantation is being practiced around the world. And there is no risk of graft versus host disease with that kind of transplantation because stem cells in essence are undifferentiated and unspecialized. So they can go into your system and they're not going to produce that rejection. So my question is, what is different about the kind of stem cell transplant that you have presented here, the allogenic stem cell transplant, versus that other kind, and where does the rejection, is it the hemopoietic variety of the cell? Like where does rejection play in to what you've presented?
Gotcha. So the hematopoietic stem cells are the stem cells we're talking about in this process, so not embryonic or other stem cells. So those are the ones that we need because we're trying to capture the cells that can divide, can multiply, and have the cell that we need from that myelomonocytic line to get into the CNS. So I'm not sure specifically what stem cell transplant, whether it is autologous. So umbilical cord, and I know Dr. Prasad is going to speak to us a little bit about that today, but when you use umbilical cord or any allogeneic hematopoietic stem cell, I don't know of any transplant in that regard that doesn't have the risk of graft versus host disease.
I'm pretty sure there is.
So,
in fact, I think Justin Freedom is
auto.
stem cell.
Okay.
Mesenchymal cord
So
tissue.
different.
They can go into anybody's system, and I know
Sure.
this because I've witnessed. I know that, there's no risk of the rejection, and that's kind of one of the beauties of this
Sure.
entire science is because they're undifferentiated and unspecialized.
But then I understand, hemopoietic variety of cells, those would be a little bit more specialized and thus present maybe potential for rejection. I get that. And then, you talking about the autologous and allogenic, that of course is interesting as well.
Right, so and I'll give you, Vinod is jumping, go ahead.
Yeah, so I'm Vinod Prasad. I'm a bone marrow transplanter at Duke. And I just want to talk a little bit about the mesenchymal stromal cells in the context. So there are, including our own center, is looking at mesenchymal stromal cells derived from cord tissue for the treatment of various diseases. But the issue when you want to do a transplant for inherited metabolic disorders, whether it's ALD or MLD or Krabbe or any Hurler, for example, is you want these cells to be present in the body, in the bone marrow, and to produce and continuously supply the stream of cells, microglial and other cells, into the brain and potentially peripheral nerves for the rest of your life. Right. The mesenchymal stromal cells don't have the longevity to stay in the body forever. So while they do not give graft versus host disease and they don't get rejected in the traditional transplant sense, they don't have the ability to live forever, and that's why we can't do those kinds of transplants.
So then what you're suggesting is that with this variety of transplantation, there is the potential for long-term.
Absolutely.
That is obviously very important, the difference between 1 month or 5 years.
We're talking the rest of the life.
Yeah, I understand that. Okay, that's interesting.
And then the auto piece versus the allo piece. So allo for transplant is you're getting cells from someone else. So there's always this potential. Auto, which is what's being done with gene therapy, is your own cells are being collected and used, so you don't have that risk of graft versus host disease.
And then this is a theoretical question about the autologous variety of transplantation, is ALD is fundamentally a genetic disorder mutation. And so how is it that an autologous thing could work when you're taking out your own broken cells and putting them back into your body?
So it only works for gene therapy. So if we were to do a standard transplant with your own cells, it doesn't do anything. So replace the modification to provide you with the functional DNA material. And so that's what's happening here. And so the beauty of it I see from GVHD is that GVHD can be a life-threatening, devastating complication. And so if you are using your own cells, that risk of GVHD isn't there. And so it's really one of the things that's really appealing about gene therapy compared to allogeneic stem cell transplant using cord bloods or other cells. That's great.
Hello. How long will it take until the FDA approves this process here in the United States? And then my second part question that may or may not be relevant to this is CRISPR. Does that have any type of relation to gene therapy, and did that help advance the current study that's out there? Because I know that's a relatively newer topic.
So the answer to the first question is really hard. So I know that Bluebird is ongoing conversations with the FDA, and it's hard to pin a timeline down for the FDA, so I don't think I can answer that one satisfactorily.
I know that, but I just look at earlier you had an 88% success rate, which obviously is outstanding, so thank you. But there's kids and people that are suffering. So at what point do they look at it as a mean to justify the end?
So it is hard to say, and I would hope, and this is based on no inside FDA or Bluebird or any information, that within the next few years, I would hope within the next 5 years at the long end of things, we would have this availability, and our goal would to have that much sooner than that. I think the data is very compelling. We have the additional cohort. I wouldn't be surprised, and again, this is just my impression, if they want to wait to see the additional cohort data mature a little bit. Gary?
So we are in conversation with the FDA right now, and what we've negotiated with them. So as you can see, 17 patients have met the primary endpoint. The full trial is 30 patients, and with the primary endpoint being 2 years, we have talked to the FDA about the severity of this disease, the critical need for patients. Is there a way that we could file prior to the full cohort maturing? And so they have agreed to enable us to file in 2019. And so we are planning to file in 2019. Exactly how long the approval process will be is something that we can't control. A year, probably.
Right, and I think
2020, exactly. And we do have breakthrough therapy designation with the FDA, which typically enables a stronger partnership with the agency. And so hopefully that will help with this as well.
And one of the things that's interesting about the FDA piece, so I'll get to that one. So the interesting thing about the FDA is that this is new. So even trying to figure out what the FDA does for a drug compared to what they do for cellular therapy is really different. So there's not tons of precedent. There's some with CAR T cells and other therapy. So I'm hopeful that this makes us move faster, but I think we just don't know. And as far as CRISPR, what CRISPR technology is a gene editing technology, as you know. And so I don't know. I think it is appealing for ALD. I don't know of work that's been done doing gene editing specifically for ALD at this point. And so where people go as far as gene therapy versus gene editing, I'm not sure we know how that's going to land.
Isn't gene editing gene therapy? Like, isn't it the same thing? You are correcting one's own DNA structure.
So you're correcting it in different manners, and so we're using enzyme therapies versus viral. So they're all a part of the same spectrum, you're right. But there are specific trials using CRISPR. So CRISPR is a very specific enzyme-based gene editing technology. And what's interesting about gene editing in just big pictures is that you can add genes, you can take genes away, you can change that sequence. So it's very similar, but I don't know of specific trials to date, at least, for ALD using that type of therapy. I wouldn't be surprised if they came. Sorry.
Thank you.
Thank you.
I think we're all aware that bone marrow transplant's a pretty complicated and expensive process. And then with the lentiviral gene therapy, it's going to be a more complicated process, and presumably there'll be an additional cost. So I'm wondering if we've heard anything from the sponsors about, like, costs, or projected what they're going to be charging patients for this and how accessible it will be for patients.
Well, so I think I don't know if costs or things. This is a tough one. Have been assessed, and there's no doubt that the cost, personal opinion, the cost of gene therapy and that transplant will be very different than a stem cell transplant. Just for baseline information, an average stem cell transplant, if everything goes smoothly in the United States, is around $350,000. If things do not go smoothly, that can go up very, very quickly. So I think that is also part of ongoing conversation with not just Bluebird Bio, but also with payers, with Medicaid, with all of those agencies trying to figure out how to best address this new technology. There's been a little bit of work done with something called CAR T-cell, which is a cellular therapy, which is simply new in patients with leukemia, to try and figure out which pathway we're going to go. And so hopefully that will help, but I don't think we have clear data about that. I'm hopeful because of the severity of the disease and the data that's been shown, and once FDA designation occurs, that the payers, the insurance companies will jump on board and support this, because it really can be sort of life-changing and a whole new paradigm for treating boys with this disease.
Hi, so I was just wondering, what was the average LES score at the point of entry to the trial for the 17 or 19 boys?
Let me pull it back up. I think the average was 2. Thanks, Mike. So the average was 2, and there was a range. They did tend to be on the lower end. So to be in this trial, the inclusion criteria had an upper limit of 9 on LES score, so that we haven't addressed boys with more severe and more advanced disease.
Yeah, because that's the same kind of endpoint as bone marrow transplant. So just a question which I don't know if you'll be able to answer. Moving on from that, the patient 2016 went on to have an HSCT rather than continue with gene therapy. What was the reason for that? Was it time frames or?
So my understanding for that patient is that on all of subsequent MRIs, there was contrast enhancement that had changed, and the concern was that that would continue to progress and progress more rapidly, and so to try to move towards a stem cell transplant to stop that progression.
So why did they not continue with the gene therapy?
So that patient had already treated, had already gone through the gene therapy, so these are the MRIs after, which are showing progression. So had treatment, it showed contrast enhancement, so hard to say, and I don't know the specific details, so I don't want to misspeak. But at that point, I know there were lots of conversations between the family, the providers, and that was a decision that, as a team, we made.
It would be really good to have some lads, but not with high LES scores, because that would be awful for the family. Right. It would be good to kind of, I'm sure you would quite like that as well, to see the outcome of the boys who are perhaps got LES scores of 5 or 6.
Absolutely. All right, thank you everyone so much for this wonderful discussion.